Hi, everybody. So this is kind of a follow-up to the beginnings of the lecture that I put that was chapters 6, 7, and 8. This is the follow-up for 8 on genomics. Genomics is the study of massive amounts of data from genetics to distinguish species, to track ancestry, things like that. I think this is probably the biggest, most important application that we have done with genetics because it kind of makes you question, what is human and where did we come from, for example. And by human, I want to mention that I'm always going to be referring to Homo sapiens because the goal of this lecture is kind of going to be able to, or kind of going to want to get into Neanderthals and where they lie on the spectrum of humanity, right? Because we're going to see that genomics was one of the ways that we basically were able to harness and measure how much like Homo sapiens were Neanderthals, because we do have that full genome. You can look at it at NCBI, and we're going to cover in this lecture a little bit of the similarities and what that genome taught us. So I think I talk about this in the other one, in the story between Francis Collins and Craig Venter at Celera. Francis Collins represented the NIH, Craig Venter Celera, and they together created Human Genome 1, which was basically the reference genome of humanity. So in this case, this is actually data from my lymphoma studies where I'm documenting regions of copy number loss or gain between 7 different diseases. And in the top here is the consensus human genome, which we're actually at Human Genome 38 right now. And it's a much better mix of humans, basically. HG1 was actually secretly Dr. Venter's, and that was a bit of a snafu to say the least, but hey, I guess if you did it, you get it. I don't know. Probably not a good way to look at it. Um, but the good thing with the human genome is that it gives us all kinds of tools. Namely, it does so against disease. We can actually see certain regions, for example, so say for E. coli, certain regions maybe that can go on and attach to genes that are attracted to us, to our genes, for example, or can harm our genes. And the reason also is remember that we actually sequenced the E. coli genome before we sequenced the human genome. And this gave us, this kind of reinforced the idea that in humans, much like E. coli, there was going to be a gene per function, like a gene per trait. There was going to be a hair gene, there was going to be an Alzheimer's gene, there was going to be a neuron connection gene or something. And we found out that prokaryotes are much more simple and they need access to specific tools and genes for specific functions. And you can kind of see that here with the kind of the massive prokaryote here. Actually, I'm sorry, this is not even E. coli. These are two different things. This is the E. coli genome right here. This is actually the Ebola virus. I am sorry, my bad. So again, that's why I have this slide up here, Tools Against Disease, is that we can use genomics not only to look at our own genomics, but the genomics of something like an E. coli that can be beneficial or harmful, or something like Ebola. I'm sorry about that. I totally saw it. So that's the reason Ebola is so strange as a virus, is it's just massive, and it has this massive protein membrane right here. So using that, we can study, what its capabilities are. Off to a good start, right? I already messed up one thing or the other. So this is just kind of a cool sidetrack, Ebola with UNMC and how well University of Nebraska Medical Center and Emory were outfitted for an infectious disease like this. And we're kind of in an age where super highly infectious diseases have kind of become the norm, obviously. But handling this disease was particularly crazy because it's so deadly, and the hemorrhaging that it can cause by basically bursting out the wrong kind of cells and causing internal bleeding called for all kinds of different therapies. And that's the reason why we don't actually know what works when you see someone basically overcome Ebola, is because we try basically too many things at once to designate which one was actually the one that worked. But in any case, I think there was actually some uplifting news that the most recent outbreak of this has again been contained because we've gotten so much better at it. Another good example, actually a less genomic example of how we figured something out, was rabies. Again, this is something that I like to just highlight because it is a very intricate life cycle where the rabies virus is going to infect most of your brain neurons, and that's going to cause the saliva, and the saliva is how it spreads. Now, humans don't actually become they don't have the same aggression kind of phenotype that goes on in the dogs. The aggression phenotype spreads the saliva. That bites another mammal. That mammal, again, goes into that saliva stage. So the real weirdness is that people actually have hydrophobia with rabies and they don't want water. That causes the saliva and that causes the life cycle to go forth. This is also interesting. The only person to have ever come out, and that's kind of a long shot too, is a woman who got it after a bat bite, I believe, and went into a coma. And somehow the coma caused the virus to not subside, so it was still in her neurons, but it basically wasn't active or causing the symptoms anymore. So while she still carried the virus, it stopped that brain hijacking. So again, two quick examples to why knowing what you're up against can really have a good play, but that in a lot of cases with virus, it is a public health question versus a genomic response one. We'll get into how CRISPR can maybe change this someday. But I think the real key with genomics and the real cool finding, obviously what we have for medicine with the human genome is amazing, but if you look right here, this is really cool because these are two examples of previous hominid species, bipedal primate species like us. You can see the Circos plot of our genome is reflected in gray, and areas where possible contributions from Denisovans or Neanderthals are reflected. Now remember, and we'll cover this, that's a lot right there. And remember, pixels are, going to be so big. So about 40% of our genome can have potentially Neanderthal contributions or Neanderthal variants. What that means is that our genome as Homo sapiens is comprised of pieces of the Neanderthal genome. The implication of that is that, and we'll get to this in the kind of the anthropology section here, there was interaction between Homo sapiens and Neanderthals. And as you can tell, there are certain genetic regions that were actually very beneficial to Homo sapien evolution, especially as we emerged from Africa. And so, I just want you to have this time to read this. We'll actually cover some of these immune system genes. We'll cover TLRs, for example, and EPAS1. So it's very interesting to have that comparison. Okay, we've covered these before. SNPs, single nucleotide polymorphisms. What genomics allows us to do is to line up individual genomes so that we can start building consensuses. It's a big number, or that's a big letter. We can start to see that there are regions, for example, like these four, that are pretty constant among humans. But we can start to see regions that are not constant. These are SNPs. And if you start to really collect all these SNPs, you can start to see haplotypes. You can start to collect which SNPs belong to which combination. So in this case, this is in a lot of cases how we track ancestry, different types of traits, because it depends on where your ancestors came from, if you have an A in this region or a G in this region. Sometimes these changes can be found in genes, but like we talked about before, you can track changes in genes. They're not as good because the gene can actually change function then, and it can actually affect the evolution of the organism. So SNPs that are buried in non-coding regions are the most valuable because they don't, if they make a mistake here and it's a silent mutation or it's just, it's not in a coding region, then that's where you can see some real changes because you're not going to actually change, the organism's trajectory. Okay. This is it's a busy slide, but just start over here with Sanger, and then I would recommend finding the YouTubes that I've posted for Sanger sequencing and basically second generation sequencing, or what I call next generation sequencing, or NGS, and most people are going to call it NGS. So what Sanger is that you want a specific region of DNA to study it, right? And we'll use this in the lab. We still use PCR quite a bit. So what's going to happen is you'll isolate tons of copies with PCR. What you'll do is get a nice little gray primer right here. See this in gray? You'll get a primer that matches a specific sequence, probably like the beginning of your DNA nucleotide, and you're going to expose it to transcriptase that is going to basically allow the DNA molecule to start being copied. Now you're going to intermix a bunch of nucleotides. Most of the nucleotides are going to be like gray, like this. So actually I should point out the primer is just this little orange thing right here at the beginning. Okay. So you're going to put it in a pool of nucleotides, most of which are gray, and there's regular little nucleotides right here, but a lot of which are fluorophores. And we're going to see these later, even in the second generation sequencing. Fluorophores are nucleotides with a big fluorescent tag on them. So in this case, C's are tagged blue, right? Now, the thing and how we use Sanger is that what happens is that these fluorescent ones are going to stop the reaction from continuing, okay? They actually not only light up, but they're going to stop the reaction. So what you end up with is variable lengths of nucleotides that end on specific colors. And so what you'll get, now I know I'm going to go into the, I know I'm going into the text here. What you'll get is a fluorophore right here at this length, and then one more, and then one more, and then one more. And so what you'll be able to measure with Sanger sequencing is the length at which a certain fluorophore color happened. So A could be here, C could be here, this G could be here, and then the T could be right here, right? And so you can start measuring, all right, A, C, G, T. Because each time the reaction is stopped by the fluorophore, you're going to actually be able to like measure where it stopped. And you're going to be able to designate, like we do down here, what that order is. Oops. So Sanger is really slow, and you can only get away with this for about 500 nucleotides. You basically just run out of nucleotides at that point, and the chances of it going past 600 without hitting a fluorophore are too tiny, basically. So it's really accurate for sequencing little pieces of like 500. So that's actually what Francis Collins was using to map his genome, and it was like very piece by piece. So this is much more accurate, but it's slower. So second gen sequencing is kind of a build off what Craig Venter did, which is shotgun sequencing. What he would do is basically shotgun the nucleotide sequences and then map like, okay, this region, we have a consensus right here because our computers can put together, oh, look, like this match right here matches this region that we can do. So basically it's like, see how this, based on all the buildings that you're going to build right here, it's the computer's going to understand like, okay, this is the consensus region of the shotgun that you just did right here because you broke up and busted up all these. So NGS is a build on that, and it's a very important build. So in this case, what you do is you shear up the DNA, so you shotgun it basically, but in this case, you add these little adapters, and these are what are in purple right here. These adapters are usually like 4 nucleotide sequences that are going to be attracted to the 4 nucleotide random sequences of your sheared DNA. So remember, that's only, so many 64 combinations or something, right, of A's, T's, C's, and G's. So eventually you're going to bind up everything to little adapters. The adapters have a second adaptive side that binds to a plate, a little microchip. So the adapters are basically twofold. One is going to attract to, oh, not this again. Sorry. There we go. One of these adapter sides is going to be attracted to the plate chip, and it's going to stick to it, but one of the adapters has basically hooked on to DNA. So now the DNA is hooked onto the adapter, which is hooked onto the chip. So at this stage, now you send in a bunch of new nucleotides, and the video is going to do a lot better job, but I'm just doing the quick version. The video, or the, at this stage, you've got the DNA hooked onto the adapter, and then what you do is actually shear off 1/2 of the DNA, so you've only got, this is your template right here, but then you have a second adapter ready to go, and you send in a bunch of these fluorophores, remember, but this time they can link, and each time one of the nucleotides with a fluorophore is added, a little piece of fluorescent light is going to show up for that nucleotide, and your chip is specified by your computer to know exactly the region and the adapter that you've got. So from that point, it can know what sequence is being added in complement because of the complementary fluorophores that are being added at each of these little nucleotides. So your computer is going to know and quantify all these different regions. And then we can line it up. Basically what this blue right here is a reference genome. Now the reference genome is collected usually by slower and more sure methods like this. But then with something like NGS, you can go in and hybridize thousands of different pieces of a genome. And this is why today, for example, you can sequence a human genome, and especially maybe even only the exons, for less than $1,000. The Human Genome Project itself was upwards of $1 billion, so that tells you how far we've come with this. So in this case, I was going to do a little drawing, but I can tell that it's not going to cooperate very much. But if you have a consensus sequence, and then you do your shotgunning, and let's say on your plate, for example, you know, you were able to identify all these sequences right here, right? So these are all the little fluorophore informations that you got that were hooked onto your plate. And so we know these sequences now. Can you tell that we're going to be able to match, you know, SNPs, for example, that align with this read, this read, this read, and this read. So we have 4x coverage right here. That's what that's called when you see that in publications. So for this one, you have a reference, and then you're sequencing something to compare it. You have one other reference here. You're going to reference this, you're going to reference this one, and you can reference this one. So the computers can put this together and find corresponding regions, and you can basically sequence and find how much matching there is. And that's how you can compare genomes. So again, this is going to be a good time to probably meet synchronously and like watch after you've watched a couple of the videos, because they're going to do a much better job with their slick animations than I can. But the major implication is that we can track genetic changes between species. So what this slide is actually doing is tracking genetic changes between 3 different species: Homo sapiens and our 2 living cousins. Okay, so 5 to 6 million years ago, none of these 3 things existed. Okay, 5 to 6 million years ago, something existed. Okay, a last common ancestor. It wasn't a chimp, it wasn't a human, it wasn't a bonobo. Okay, it was a primate that had not yet achieved bipedal motion. We know that it was related to orangutans because they're all the way over here in the back. Okay, so they're the ones that share sequences at this jumping point. So they're the next ones over, basically. We can map this reaction because we can see changes in specific genes. So in this case, we can also use protein differences. Now remember, I told you that's a little more dangerous because changes in a progesterone receptor could actually have benefits. But in this case, it is still very good to see that in the human progesterone receptor, which is a key hormone, here are the amino acid changes. In this case, you can see the amino acid differences between chimps and bonobos too. And the reason I like this slide is that oftentimes humans are always like compared to, you know, chimpanzees, and it's like, oh, you know, like they're the closest genetic relative we have, blah, blah, blah. That's sort of true. But remember that chimps and bonobos had their own split at one point as well. So it's worth having that conversation as to which one of these species actually represents what this looked like better, right? And the best way that we can usually look at this, who represents that LCA? Do chimps, in their aggression and their brawn, they're really, you know, they're really fight or flighty, they're very angry, they settle a lot of things through, you know, death, essentially. Versus bonobos are kind of a forgotten ape. In a lot of cases, they mimic how humans usually problem solve in a lot of cases. They also really mimic our sociability. Chimps don't. Chimps are much more aggressive in their social interactions, whereas bonobos are a lot more, you know, cooperative. Most of the interactions and conflicts with chimps are solved through violence. Most of the ones with bonobos are actually solved through sex. And it's kind of a strange interaction, but it kind of mirrors that chimps may not have represented this common ancestor. They may be the split off, or bonobos could be the split off, right? And that's kind of something to look at. But if you look further down the line and you look at orangutans, which maybe are a little more, they're a little more like bonobos, but then if you look at gorillas, they're a little more aggressive like chimps. That's something that you'll kind of have to look at and decide, like, okay, what does that last common ancestor really represent? And what did you know, which one of these two does it really look like? Okay. One huge difference between the genome of a chimpanzee or bonobo and a Homo sapien is that we have a jumping gene called ALU elements. So you'll see that the coding DNA genome between chimpanzees, bonobos, and humans, there's not a ton of differences, but if you look at big structural differences, like jumping genes that insert and cause new exons to show up, like ALU, or these can come in and they can burst genes in the wrong place, or they can dose genes down because they'll interfere with promoters and stuff. These are really where we see some key genetic differences between us and any other species that we're closely genetically related to. These jumping genes are really what superdrived and really accelerated the changes that brought on Homo sapiens, Neanderthals, and the other 6 major hominid species. Okay. So for a long time, the fossil record was all that you really had to look at between, where Homo sapiens possibly came from. Okay, there was no genomics. This was completely a hunt to find where are the, quote unquote missing links. The whole missing link thing was something that was very common when I was growing up. But eventually, not only did we find, bones or tools or sites, and Lucy is right here in Australopithecus, right here, there she is. We were able to find the fossil record, but it was the genomics that we started to find here from the Neanderthals that really showed, okay, there was something before us. And that's where there was a last common ancestor between Homo sapiens and Neanderthals. Neanderthals split from Homo sapiens. Homo sapiens stayed in Africa, while Homo neanderthals went into Europe, the Middle East, and then there was another split that we'll see from Neanderthals as well. So there's even another fork for them. Now notice on the phylogeny, chimpanzees have made it all 6 million years, okay? Homo sapiens have made it all 6 million years. There's a slight dip in the Neanderthals, and that's to be explained since Neanderthals are not here with us. Okay, so let's take a look at the prevailing out of Africa theory. Okay, for a long time, people assumed that, well, for a lot of times, we actually assumed that there was some kind of major integration, that all existing hominid species simply integrated and became Homo sapiens. Now, they're not right, but they're not 100% wrong either. The out of Africa hypothesis was actually a little bit unpopular because it didn't account for some of the similarities that we were seeing in the fossils of Homo sapiens and Neanderthals. Not to say there weren't major differences. Strangely enough, a bigger brain. So what ended up actually happening was that Homo erectus would evolve, and it would go and become different things outside of Africa. But there was eventually some kind of split, a divergence, and this was Homo sapiens. And Homo sapiens, many stayed in Africa, but many went on to colonize and essentially conquer the rest of the world, essentially. And I don't want to use the word conquer without giving you some more context, which is what we're going to see later in these slides. But I'll kind of leave you to make up your own mind about that. So the challenge became, and this was a while ago, the challenge was to actually find, okay, we have Neanderthal bones, but we need a genome, okay? And this was roughly 10 years ago, and the book Neanderthal, The Neanderthal Man, is about Svante Pääbo, a scientist from Sweden, I believe, on this journey to find this full genome. And you have to understand, it was impossible when he started. That's the word that people used. Because look at the DNA that's remaining from bones, for example, thousands of years old, right? There's so little left. There's so little. And to get worse, guess what's also in this ancient DNA? All the viral, bacterial, and fungal DNA that's just been milling around while this has been fossilizing, okay? That's bad. One of the other things, this little red mark right here, that's basically chemical age. You know how things basically have half-lives? Well, nucleotides and DNA and genomic material, they start to undergo changes over time, and you have to account for these changes before you can call that, what was the natural nucleotide there. So even when he started, it was hard enough to get fresh DNA to sequence from actual tissue. And when you're starting with this like ancient DNA that's also full of all these holes, It was really hard. But one thing that he says in one of his great presentations is that never doubt where science can go in the future. You are often basically have your pants pulled down by it, as he'd say, and you'll be exposed as like, uh-oh, like you really should have waited there, you know? Okay, first breakthrough that he had was that we eventually got enough Neanderthal DNA to sequence their mitochondria. This was huge. We could compare their entire mitochondrial DNA to ours. This was the first time that we were able to look at mitochondrial Eve, for example. Remember her? She's the ones that all Homo sapiens living today have a similar, at least portion of their mitochondria related to. The reason we could define her is that the mitochondrial DNA of Neanderthals, which split far before sapiens did, does not have mitochondrial Eve's DNA in it. These are a different human sect. The way that we did this, and you can see these little strands, these were the first way to build a consensus sequence. Remember the consensus sequences that we can still build? And then this is the full sequence right here. These were made with bacterial artificial chromosomes. Essentially, just massive plasmids of circular DNA where we would have to insert and re-PCR these big chunks of Neanderthal DNA safely. That was the way to get this ancient DNA into a safe sequencing and highly replicating, basically format, so that we could start copying it over and over to get these big chunks of consensus that we could start building a consensus reference from. Now this was huge. This was a big deal, but it still wasn't the real genome, you know? This was the mitochondrial genome, but to be honest, this was an undersold first step, but you want the whole thing, okay? This was the beginning of next generation sequencing. So we had sequenced the genome by this point, but we needed a bigger tool. We needed something more sensitive because all we have is this tiny amount of DNA. So at this point too, also recollect that in the computer, you had to be correcting for all the fungal DNA, the prokaryotic DNA, any viral DNA that may have been on this, and the age modifications of the DNA. So we eventually figured out programs that could self-correct that and, you know, get rid of, you know, sequences that were clearly fungal or something. Or you had to get rid of sequences of current human DNA. So if you touched the bones, for example, your DNA might be on it. So the amount of careful care that had to go into these bones was immense, more than you would imagine. But NGS, what it allowed with that big like chip structure like I showed you, it allowed a tiny piece of DNA to be sequenced tons of times to get huge consensus on, a straight strand like this. And we could have tons of clones, and the computer could find us what's the actual sequence of this Neanderthal segment right here. So you could take a piece of DNA and extract it from bone. You would apply it to these libraries, and they would self-replicate over and over, and you would read that chip. You would insert this into the sequence database of humans, and you would start comparing it, because at this point we had our own genome. We could start aligning nucleotide sequence, and alignment is the key here, because we wanted to see the differences between the Homo sapiens and the Neanderthal genome. Now this was meant to ask a very big question. As Homo sapiens, because Homo sapiens first what they did was they took over all of Africa. And Homo sapiens is us. We took over all of Africa, right? As Homo sapiens emerged, we wanted to know that if there was an interbreeding event, we would expect in this case that Europeans would share more Neanderthal variants than Africans who would stay here, Homo sapiens that would stay here, okay? We did find that. What we find, and even to this day, if you have ancestry from this region, you are more than likely to have more Neanderthal variants inside your genome than if your ancestors are from this region. And this was a key finding. This is something that had only been surmised. We couldn't tell it from fossils, but we can tell this from genomics. We know that Europeans share more in common with Neanderthals. What this means is that as Homo sapiens came out of Africa, there were interactions, Neanderthal and Homo sapien interactions. And that's We're going to see how that started to become a little bit messy. So one of the keys that allowed an even more efficient method was, in the old way, with the libraries, you had to double sequence. You had to keep the full double sequence intact. What new NGS did was actually split this. It actually split these two sequences right here into two, so that you would have two probes going at once, because you would just self-replicate one side here and one side on this side. So you would have 2 times the amount of sequencing. That's just kind of a cool little step, and like kind of an intuitive step. It really makes you think of how cool the engineering, the chemical and genomic engineering really goes into this, and you really see DNA as this really cool structure. But this was another key step that allowed even more coverage to happen on the Neanderthal genome. And what that allowed us to see was that there were tons of regions that were consensus Neanderthal genome regions that could be found in Homo sapiens today. So on average, although you only see about 1 to 2% of a person's genome coming from a Neanderthal, up to 40% of the Homo sapiens genome can be traced back to Neanderthal. That means that 40% of the genomes of Neanderthals collectively, so in all humans, still exists today. So even though Neanderthals as organisms don't exist today, a large, substantial portion of their intact genome exists as a whole in humanity, in Homo sapiens. So it was a huge finding. And he makes a joke right here that when they found this, a lot of people would email and be like, I self-identify as Neanderthal. And it was tons more men. But what was really funny was that women would call and say, hi, I think that my husband is a Neanderthal. Please sequence his DNA. I'm pretty sure he's a Neanderthal. So it was pretty funny as this started coming out. It was like, oh boy. And it was an amazing find. But even more so. So here's Homo sapiens over here. There was something else. We started finding from sequencing some of the other bones, those from Asia and Oceania, that there was another split. So when Neanderthals all that time ago split from Africa and had their own out of Africa, and they ended up throughout Europe, throughout the Middle East a little bit, there was another split. And that is those Neanderthals, or to be Neanderthals, so Homo erectus is actually right here, that went to Eurasia, Asia, and Oceania. These are Denisovans, because we started sequencing human cousin remains and bones from these regions, and we found that they were genomically distinct from the Neanderthals. So there is a second consensus species that we found. And this map does a good job showing where the highest concentration of those genomics are still found today, specifically in Oceania and the Aboriginal populations here, and in kind of the Asian area and the steppes. So this was very interesting and exciting. We knew for a long time that Homo sapiens basically got rid of they basically took over Africa, and then there was an emergence. It's interesting because we actually have evidence that there was an initial push here, but that Neanderthals actually pushed Homo sapiens back. And we'll see what changed the second time around, because eventually what we see is that Homo sapiens started to distribute, and we can map this genomically. The other thing that I'll tell you is that these points of contact here that you see in exclamations, these are points at which Homo sapiens would start to show up, and fossils and artifacts start to show up. They're also the same exact time that fossils and artifacts from Neanderthals in blue and Denisovans in red started to disappear. So when Homo sapiens would come, you often saw the other human species go away. Additionally, you would see some people attribute this to climate, but pretty much wherever you look, wherever Homo sapiens went, 90% of large mammals would be eradicated quickly. We'll go quickly into the interdisciplinary side of what made us so different, to say the least. And the other big conclusion here is that we did find that there was intermixing between quote unquote species. And I don't want to, I want to use that word lightly because genomically, yes, we were very distinct, but we had fertile offspring. Morphologically, we were very, very similar looking. So it's really up to you how much, where you draw a line on the word species, right? Or were they human? That's where the gray area of science is so intriguing and so sometimes troubling too, in this case. But as you can see, there were lots of events where Homo sapiens would come into contact, let's say right here with Denisovans, lots of times where there was contact and interchange between Homo sapiens and Neanderthals. And strangely, right here, there was a lot of contact between, there was some contact between Neanderthals and Denisovans. And that happened right here. Strangely enough, there is a bone sample from a cave here that was a mix, 50-50, of Neanderthal and Denisovan genome. So a hybrid. And again, I don't even know if that's even the right word, or if we all collectively truly are just the same thing and all this is for naught. But the genomics showed that, strangely, one of the fossils that we happen to have is a child between a Neanderthal and a Denisovan. So, getting into some of the nitty-gritty of what was given to modern Homo sapiens by Neanderthals. One is the gene SLC16A11. Okay? What this does, actually, unfortunately, is a risk factor gene, and it's a distinct allelic variant that has Neanderthal origins that was carried over when the crossbreeding between Neanderthals and Homo sapiens happened. This actually makes you more susceptible to type 2 diabetes, where your body basically it ignores the insulin and it just piles up bunches of sugar. That's a bad deal, unfortunately, for disease, and that's why you see in some cases Westerners or people with more Neanderthal influence in their ancestors develop type 2 diabetes a lot. The interesting thing, and so this is like the strict, here are the specific Neanderthal changes that other Homo sapiens don't have. It's predicted that this variant was actually selected for in crossbreeding individuals that had to undergo periods of starvation. So that's actually kind of interesting. It's like, why would this be a disease thing? It's like, well, the best reason we can find is that these variations actually make it easier for you to last during starving times. Basically, ignoring insulin during those times apparently has some benefit. So that's kind of interesting, and it helps us put together what human evolution and culture may have been like, especially through these thousands of years of transitions. Because remember, millennia are passing during this time. Denisovans have less direct contributions, but one of the coolest contributions that they have is a variant in the gene EPS1, and this can be found specifically in people from Tibet, or those of Tibetan ancestry. It's highly enriched. Look at the incredible frequency. 80% of Homo sapiens from Tibet carry this variant, and the reason is that people with this variant have a natural disposition to survive high altitudes. And so this is actually evolution in real time. As Homo sapiens broke into very high altitude places, like the Himalayas, they likely encountered Denisovans. There was likely interchange and crossbreeding, and those that had the EPS1 variant had a better survival chance than those that didn't. It's a very interesting, very quick, accelerated look into these genomic contributions that these species made, with us, essentially. And I keep using the word, but it's really, again, for you to decide what the separation is. But the greatest contribution in a lot of cases, innate immunity. Now, we've talked a little bit about this, but what you have on a lot of your immune cells are toll-like receptors. That's what this acronym is. A lot of your immune cells just patrol around and they look for these things. They look for stuff that does not look like human, okay? Something like that is going to be like double-stranded RNA. That's a virus. Lipopolysaccharide, that's like a prokaryote that's invading you. Single-stranded RNA, virus. Flagellin, bacteria. These receptors go around on natural killer cells or some of your other innate immune cells, and they just bind to these targets and they kill that cell. They start killing stuff, okay? They're great immune defenses. We got TLR1. So TLR1, sorry, I'm trying to grab this one. Oh, well, never mind. TLR1, TLR6, and TLR10 are direct contributions to Homo sapiens from Neanderthals. Neanderthals developed these during their specific evolution, and then during the genetic interchange, Homo sapiens ended up with them. Specifically, those that would settle in the European region, Homo sapiens, ended up with more of these because there was more interaction in that case. That actually strangely made European Homo sapiens very much more immune to certain types of, or ready for certain types of diseases. We see this later in history when Europeans will show up to places that do not have the same immune capabilities. We also, and this is a whole biocultural story again, things like smallpox, influenza, those diseases started really showing up once we started domesticating animals and bringing along piles of animals with us, just huge herds. Before that, viruses or things like this didn't have a pool of infection in which they could evolve, in which they could dive in and infect bunches of things. So between agriculture and some other, basically evolution against disease, certain regions of Homo sapiens started to have specific innate immunity capabilities. And eventually when you would have, new interactions like Europeans did in some cases, those interactions would not be beneficial for anybody who had not seen that type of disease, specifically the diseases that circulate in farming and mass animal cultures. This is one of the strangest things. So remember, we've talked about coding changes are good, and we do see a lot of coding changes. So here, this red threshold is basically saying, this is a high amount of, exchange. So UTRs and protein coding and, areas over here, a gap between genes, those are big regions where genetic change was found between Homo sapiens and Neanderthals and Denisovans. It's so strange because Homo sapiens kept a lot of their promoter and enhancer regions. In fact, with promoters, it's basically like we didn't borrow any promoter regions from Neanderthals at all. Homo sapiens kept a lot of our epigenetic elements. We took on a lot of new protein coding genes that were like, hey, when you evolve with this, you do better in the cold. When you evolve with this, you do better starving conditions, right? But a lot of the epigenetic factors that separated Homo sapiens from Neanderthals stayed. And for this reason, it's theorized again, and it adds to the evidence that the advantages that Homo sapiens have, specifically with our intelligence and our sociability, they are epigenetic differences. They are on and off differences. They are dosage differences. They are not so much that we are genetically so far apart coding. It's really that we kept, the specifics that make our genes go on and off in the right pattern. So, one of the key things that if we're looking at a split from, let's say, Homo sapiens right here and Neanderthals, not only can we see, what are the contributions over here, that's all fine, but what are the things that evolved and changed to make sapiens essentially superior to Neanderthals? Because if we look back up at that map, there's not a really good way to say that Neanderthals had enough advantages over sapiens to win. And I don't know if winning is the right word, but we'll follow this up at the end. There were 6 hominid, walking, tool-using species of humans, essentially, when Homo sapiens emerged in East Africa. Today there is 1 Homo sapiens. So what I want you to highlight from these genomics is what we found out from the Neanderthal genome is we started to find out regions that we had that they didn't. These are 3 genes, for example, that are highly active in Homo sapiens. Strangely, what they do is they control and modulate metaphase of neurons. And that's what you're seeing here is a neuron dividing. What they do is they make this process go slower. They make this process have a lot more control. And what that means is that our brains have a high plasticity. Our brains can change very easily. That's why we can learn. That's why you can teach a Homo sapien a lot of different things. That's why Homo sapiens can go back and strategize. It's because our brain can adjust. The neuron pathways and the information that we exchange can change based on our environment. That's why we are the species that we are. Is you can drop Homo sapiens in any environment and it can start engineering the environment towards itself. Another massive thing that we saw, so this is conservation. So in red, you'll see regions that are conserved between Homo sapiens and Neanderthals, so those don't change. But there are a lot of regions with this RNA right here that we're looking at. It's a hairpin RNA. It's a 3D RNA gene. So remember how we said RNAs, proteins do most of the work? This is a very strange gene, HAR1, Human Accelerated Region 1. This RNA has a lot of enzymatic capabilities and regulatory abilities to dictate our brain development. And it is very different from the sequence that we find in Neanderthals. So this small 3D RNA is responsible for a ton of our brain development. And even the smallest changes can have massive effects, as you've seen, especially when you consider how it may affect the 3D, basically the 3D shape of this and what that's going to modulate. Some of the regions, so this is neurobiology we're getting into. So remember, this is still controlled by epigenetics, genes, things like that. So remember, this can change genomically. And these regions, these A through E right here, these are the brain plasticity, physiology, and cell biology that makes our brain able to adjust. It makes us able to prune old knowledge to make way for new knowledge, new strategy, new creative ideas, right? Here you can see new cells forming, for example. You can replace specific myelin segments. And this, so actually what this figure really is showing is that the way that our brains are so plastic is that we can manipulate the myelin sheath, which is basically that neuron insulator to keep neurons stronger or weaker. We can make it thicker, for example, as we do here, so that this connection becomes stronger. We can lengthen connections, we can shorten them. These nodes of Ranvier that are always going to be in between these little pieces, they can get shorter or longer. This is how memories and knowledge are circulated in the human brain, and Homo sapiens have it better than anything at adjusting the brain. That's the thing. We are born with very little instincts, and we found out that Homo sapiens are built basically to run this show because every time we come up against a new challenge, we could re-strategize. We could always overcome whatever the environment threw at us, and that was the special thing. Now, Neanderthals actually had a bigger brain, but they didn't have the ability to change it that Homo sapiens did. And that's where we start getting into this story. And this is the last, so this is the last part of the slides. If you're done with genomics, you can log out. This is just kind of going to be the story of where this finishes. So again, we covered this. Homo sapiens emerge somewhere around this time. Homo sapiens try and basically get in and out of Africa. Couldn't draw an arrow there. And the spread starts. And you can start to see that certain regions were only occupied so long ago. But in a lot of cases, most of these migrations happened, or at least started in this time frame. Okay? But the question up here is still posed. There were 6 species to start. One is right here, and it's Homo sapiens. There were 2 others in Africa, and there was one in Oceania. There were Denisovans here, and there were Neanderthals here. So that's 1, 2, 3, 4, 5, 6. At the end of the day, though, so if we just look at those species. Okay. At the end of the day, though, when Homo sapiens emerged as what it is today, and we'll talk about why, these species left, they were gone. These species eventually were all gone too. There was only one. Now you have to ask yourself at this point, what happened? A lighter version of this story is that Homo sapiens were able to strategize and hunt better, and thus we just took all the food and out-competed our cousins. But there is the other option. which is when things move in, when life encounters other life, there is conflict. And that conflict, in that conflict, those with the advantage will likely succeed and take over. Now, I'm not here to give a history lesson, but the likely scenario of what happened, knowing the Homo sapiens nature, we know that we're not the nicest things on the planet. But this is always a good quote. I'll let you pause for a bit here and kind of consider what we've talked about when it comes to the word species, right? Other human species that existed with us. And as we came out of Africa, when we encountered them, what history might have looked like had Denisovans or Neanderthals survived. I'll let you pause here, and we'll move on. Okay. So like we talked about, there were a lot of these interactions. And as we can tell, with modern humans, there were a lot of, basically good interactions. There was a lot of the time where, there was a coexistence, and we know that from the genomes, from genomics. That's the power of what genomes can give us, is a look into this past and a look into what makes us who we are. And I think that's more of a power even than medicine sometimes. This to me is more powerful and earth-shattering than medicine in a lot of cases. But even as a genomics person, I do want to caution you, trust me, Homo sapiens are a very, very special thing, okay? You can't discount us as, okay, that's it. They are what they are. We're just another animal, or we're just another, whatever we are. We're just floating in space. We have the ability to study ourselves and make a better world. And that's, I think, where that importance comes from. And again, this is also a very good look at what I was kind of talking about before, where bonobos, and then you have chimps, and it's a very good idea to look at the behavior of bonobos and kind of think like, okay, like maybe this was the common ancestor, and this common ancestor was a more hypersocial thing than the hyper-aggressive chimps that maybe split off, for example. But, oh yeah, I was wrong. Gorillas are more related than orangutan. But if you look at gorillas and they're kind of turned to violence, it may be that this ancestor was more like this ancestor and violence was more in our nature, and we kind of overcame it. And bonobos were kind of a more specific split towards a more social, like sexual culture than a violence culture. Okay, again, like I said, this is all green text, so this is all mainly for fun and genomics, but here's the big thing, is that what gave humans Homo sapiens, a special ability over the other species, is that we could form groups over 150. Mammal groups break down at 150 because you stop knowing who's your your family isn't as big in the group anymore. You start breaking those kinship, those friendships and stuff. So even chimp tribes or even bonobo tribes start breaking down at 150. And it's not because of intelligence, it's because of sociability. It's because of fear of one another. And that's the big separation between Homo sapiens and even Neanderthals and Denisovans, is that Denisovans and Neanderthals could only form groups of 150. And Homo sapiens don't get our power from the intelligence of a single organism. We get it from the collective strategy of a massive group working together, okay? And the ability to work together and be social had to mean that we had to lose a lot of the genes, even if it was epigenetically, for the fear response. Chimps have aggressively high amounts of fear and stress genes in their neuroendocrine pathways that humans basically have turned off. Okay, because if chimps were in this close of proximity all together like this, fights would break out, violence. Even with bonobos, it would be very uneasy. But humans, for a long time, we've been able to do something like this, and it's amazing. And although we had big brains, Neanderthals had bigger ones, and they were also way more physically able to fight. That's why Homo sapiens got beat back the first time they tried to come out of Africa, but we re-strategized and we got a bigger group and we came back. That's the point, is that humans and the human brain and the human tendency not to fear other humans, that's the big reason we got here. Because you can build culture, you can build ideas, you can build stories in social structures that a single Homo sapien can't build. This was the big difference in our brain, was that a massive swelling neocortex able to think and strategize of things that didn't physically exist, that was the key. And again, remember, we're in a genomics class, and I wish I could go more into genomics of neuroendocrine, but think about that's where that myelin sheath stuff is coming in, and this is where the genomics that make Homo sapiens different was way turned up to 11, basically. What happened is that our brains started to become so big with this social interaction that we started to be able to think of things that didn't exist yet, okay? That means you can think of strategies. You can think ahead to the future, right? You can say, the Neanderthal band that we were just up against, they beat us back, but we can try something new. We're going to come back together and we're going to re-strategize. And strategy is still how humans can beat massive, deep AIs at games like StarCraft that are really complex, because multiple humans working together can always still strategize together better. One of the keys to that was language. We know that the gene FOXP2 is vital for language. It's even vital for bird songs, right? Isn't that crazy? Homo sapiens have a very, very big set of intricate differences between our FOXP2 and the FOXP2 of other human species, okay? Now, the first human to have a mutant FOXP2, yeah, maybe they had some good capabilities at that point, but it's not like everybody else had FOXP2 at that point. So a lot of the biocultural argument that shows up is that to survive in a band of Homo sapiens, you had to learn language quickly. And that's where a phenotypic plastic brain, one that can learn something that you may not have the capability to be born with something, that's where this argument and the genetic arguments start to kind of wrap around each other, is that yes, we have genomic regions that code for language, but culturally, we had the social ability to teach language as well. Now, as far as we can tell, FOXP2 eventually emerged as such a clear advantage that I think all humans today have the variants that we have to understand language, but it was still a very Lamarckian, will to survive to rewire in this case. And this is where we get to why Homo sapiens have the advantage to this day, and why they had it in the conflicts that followed the emergence from Africa, is that the reason we could form more than 150 groups was that we could create things or have ideals that did not physically exist that we could all follow at once, right? This is what gets groups together, bigger than just your family and your friends. And the beginning of this was animism. This is animism. This is the idea that everything has a spirit that you cannot see, but it exists. Okay? And trust me, when we're going through these things, in their own way, they do exist. Truly. I do mean that in every sense of the word, because of what a Homo sapien brain is. And we can go into this world of intersubjective. Intersubjective is something that empirically we can't find or measure, but it does exist because of our dependence on culture and sociability. So the first thing that united all these large groups of Homo sapiens in a lot of cases was animism, was that we follow the spirits of the world. We follow the spirit of the world. Eventually, I know this is a genomics class, but this is the final finale for this. Eventually what happened is that there were forces that were non-living that Homo sapiens started to convene with, make deals with, tell stories about. These became the polytheistic gods, okay? These were, and we still see polytheistic religions, but a good example is, the Greek and the Roman, stealing of the Greek religions. Basically, you had all these forces like rain, wind, fire that you really they didn't have a spirit, they were intangible, they weren't living things, but we still characterized them as something. And you could still commune with them, and you could kind of make deals with them. And that's why in a lot of these cultures, these polytheistic cultures, they're very, there's lots of personality behind each one, because each one had their own kind of story. Now, what they're surrounded by in this picture is the universe itself. There were forces of the universe, and in Hindu, even to this day, there is a force of the universe. But you don't exactly commune with that force of the universe. You commune with all the other, or the Greeks would commune with all the other gods and make deals and, things like that. You can't just go up to the universe and say, I would like, this is what I would like to happen, right? That's where monotheism started to take over. Monotheism was where groups of Homo sapiens started to say, instead of turning to the living or the non-living forces of the world, I'm going to turn to the universe itself, the everything. And that's who I'm going to commune with, and it should be who you do too. Now, a key difference here, and this is, a lot of this is borrowed from Sapiens, a key difference with what happened with monotheists is that polytheism didn't really breed a lot of warfare and stuff between different religions. So it's like, well, you have your characterizations, I have mine. When you would have two different groups that had different monotheistic ideas, the problem becomes that I pray to the mass universe, the whole universe, so yours can't be the same one. That's where a lot of these problems started to show up with humans. And we're well past Neanderthals at this point. We're into history now. And then a lot of the cases, the monotheistic version of the universe that we could commune with started to be humanized. Monarchies, popes in some cases. And this was the intersubjective, but then bringing it down to the world and saying like, oh, we actually have, something tangible that we can put this on. Now today, we even have intersubjective ideas that we follow as well. The idea of liberty, justice. You can't see or measure justice, but in human culture, it is something real, right? And this is still something that unites humans. These ideas that our brain and our sociability can create and build and, basically make amazing things with, And we are on to a new stage. People tried this before, but they bastardized it. We're on to a new stage of playing with the blueprints that make life. And this is where things are going to evolve and change in the next, 100 years is we are going to start changing the blueprints of life. We've already started making, synthetic vesicles that replicate. We've already changed human DNA. We've already inserted genes. This is why genomics, I feel, is going to play more of a cultural role than we think in the future, because we are going into intersubjective ideas that we can build as a social structure and a social species, but we are changing fundamental, basically chemistry about what we are as a species, and that's where our next evolution may actually happen, is that we may augment the changes that we always thought kind of took for granted. So bit of a longer one, bit of an interdisciplinary lecture. So if you hung on this long, definitely let's have a fun discussion about what you agree with, disagree with, because like I said, I'm kind of just presenting this. Don't take what I say as an endorsement. And if I miss stuff, obviously I miss stuff. We're human. So you can definitely let me know, and I hope I didn't, but obviously that definitely can happen. But I love discussing these ideas, and I love discussing how genomics can inform us more about who we are and where we're going, more than just the pure science of it. So hopefully you're as excited about that as I am. But until next time, have a good one, and I hope you interact with some humans recently. I know we're still quarantined though.